Our special senses, we're only going to cover a couple. So we're just going to do sight and hearing. And we are really just covering this basically. You can get a whole degree in either one of these, right? Anyone interested in actually pursuing, let's say, ophthalmology or audiology? No? Okay. Well, you could. Let's see. So there are other special senses. So, of course, the touch, pain, pressure, and temperature in your skin. And let's see, the taste. We're not going to go over that one either. We'll actually probably cover that more when we do the digestive system. All right, so the parts of the eye, however, you should have a decent grasp on, right? Because we covered this in lab as well as the ear. But again, we're going to add, all right, well, what is the significance of these structures? What do they do in sight? So we'll start with where the light enters the eye, which is the cornea. Okay, and the cornea is clear. Okay, but it extends from the white part of your eye, the sclera, which is really just the fibrous connective tissue which is holding everything together. Okay, the cornea, as light passes through your eye, it is controlled by the iris, which is smooth muscle, okay, which is controlled by your autonomic nervous system. Right, you don't control how big your pupil gets, but that's the interior, but then part of the iris is the pupil. Yep.
In the video lecture, it said that the iris only contains brown pigment.
Right.
So how does that work with like different colored eyes?
So the pigment, so yeah, if you were to lay it down, dissect it out and lay it out on the table, it would just look brown. It all look the same.
People?
What's that?
Like even blue-eyed people?
Right. So what happens is you have fluid. Okay, so this is filled with aqueous humor. And you do have structural differences in the iris, which diffract light at different wavelengths and thus produce the different colors. So you can see the same thing actually in bird feathers. So birds, some of them are actually red. Some of them have like a multiple, like if you ever look at these starlings in the spring, they have like kind of a rainbow sheen to them. That's because of the microstructure of their feathers. Depending on what angle you're standing at, produces a different color. So it's similar with your iris. So there's also something that was mislabeled, I think, on your video lecture. You have two main parts of your eye. The anterior cavity and the posterior cavity. Okay, the posterior cavity is everything behind the lens, and this is filled with vitreous humor, which if you remember when we dissected the eye open, it was like jello. Okay, so it's a thicker fluid. And the anterior cavity is filled with aqueous humor, which is more liquid, less gel. Okay, the anterior cavity is split into two smaller chambers. So from the iris to the end of the cornea, this is the anterior chamber. And then from the lens to the back of the iris, or the back of the iris to the lens, this is the posterior chamber. So this is confusing. And that color isn't very great. Let me write with black. So you have an anterior and posterior chamber, you have an anterior and posterior cavity. And then there's some other structures which connect these, and the flow of fluid between them is very important. If you have a blockage, it can cause problems in your eye. But we aren't going to go over those microstructures. Okay, so behind then our posterior chamber, or at the end, we have our lens, which is being held in place by the ciliary body and attached by suspensory ligaments. And the ciliary body is another muscle which contracts and changes the shape of the lens to focus light at different depths on the back of your eye. So light goes through the cornea, through the lens, and then will project an image on the back of your eye. Okay, we'll go through some of the layers. So under the sclera, we have the choroid, which is the vascular layer. So the blood vessels, the larger ones, are in the choroid. And then on the inside of that, you have the retina, which contains your photoreceptors and the corresponding neurons which transmit it to our optic nerve. Now, there is a part on the retina where light focuses. Okay, the larger part is called the, macula lutea, sometimes just called macula. Yeah. But you also have a very, very small part in the very center called the Fovea Centralis. And this is where light focuses. This is where a focused image is projected in the back of your eye. So whatever you're actually looking at, which is in focus, that's because your eye is positioned in a way to where light, that image, is focused right there. And if you have vision problems, it's because the focus is behind or ahead or in front of that area, and you see everything blurry. All right, so what makes this different, though, from all the rest, the Fovea Centralis, is it has, so all of the retina layer has blood vessels running through it, so going to all the different cells which are in there. But at the Fovea Centralis, it does not. So there's no blood vessels blocking the light and the image as it is projected there. And then you also have ganglia, so these processing clusters of nervous tissue, which are directly attached to the photoreceptors. And in all other areas, the ganglia are shared among many photoreceptors. So we'll talk more about that in a minute. But this allows for more information to be processed and nothing is blocking it. Okay, so those are the parts of the eye. You are very familiar with the muscles, right? All right, so which changes the direction of the eye. We have our rectus muscles, which I've drawn already here. And let's say this is a right eye. So this would be the medial rectus. So this eye, turn it around. And this would be the lateral rectus. And although I've drawn them like right on top and right below, right to the left, the superior and the inferior rectus are actually off by a little bit. So they're off by like 20 degrees. And so when they pull on the eye, they actually pull on it at an angle, okay, which makes it, so if you look up, it's going to pull at it at a weird angle. So to correct for that, you have the superior oblique. Oops, spelled that wrong. So when the superior rectus contracts, so does the superior oblique, and it rotates the eye to correct for that slightly off pull as you look up. And then likewise, when you look down, the inferior oblique rotates in the other direction when you look down. And essentially, this just allows you to stabilize your eye when you look up or look down in any direction. It's going to tweak it a little bit so it holds it in place. So. Well, what happens is there's wait, so what was your question?
So both the superior and inferior oblique, they both go inside, they both go medially?
So what happens is actually on the superior oblique, there's a ligament here that pulls it back, and then it comes across there. So when it contracts, it pulls it medially.
Are we right or left eye?
Right eye.
That's a right eye.
Yeah, both rotate the eye immediately.
Well, it's clockwise and counterclockwise, right?
I guess, yeah, because that's not the right way to ask the question, I guess.
Yeah. So it's pulling it medial, the other one's pulling it down away. But yes, yeah, they are both medial. But it's clockwise and counterclockwise. So if you look where it's positioned, if it contracts, if the inferior contracts, it's pulling it that way. Yes. Okay, so the handout has you label all these and then also gives you space to define, all right, what do these structures do? Okay, which we just went over. All right, this gives you a little more, this is from the book, a little more on the structure of the retina and how the photoreceptors work. And so it's really interesting. What you see here, so the ganglia cell, zoom in there. So in this area, you have bipolar cells, which are transmitting information from the photoreceptors. So these green and blue, funny shaped cells at the top. And they are transmitting the information to the ganglia, which is going to process the information and then send it on to the optic nerve. Okay, and here you have almost a one-to-one connection. So this would be something that would be similar to where you found in the Fovea Centralis in the macula. But in other areas, so actually probably more accurately, you would have one ganglia sharing or being shared among up to 50 different photoreceptors. Okay. There's two types, there's actually 4 types, I guess, of photoreceptors. The cones are for color. All right, and we have red cones, blue cones, and green cones. So what you learned in primary was not correct. The primary colors are red, blue, and green. But we don't have yellow cones. But among those 3, in combinations of those 3, you can create the visual spectrum of what you can see. Okay, so these are concentrated at your fovea centralis in your macula. That's the area where you want the most, precise vision and color. And they work in intense light frequency. So if there's lots of light, these guys are working. In low light situations, you have rods. Okay, where's our rods over here? And these only have one type of photoreceptor, and it's rhodopsin, and that's what's shown over here on this side. But these are just the absence. They have a binary presence, absence of light. Okay, so they tell you these work best in low light situations. They're working the whole time, but you don't really notice them until you're in low light situations. And your color vision almost disappears in low light situation. So when it's twilight or dark, you just kind of see images similar to the tongue camera. You can't really see colors. All right, and the way that they work, so they are embedded within the plasma membrane. They're transmembrane proteins. And they have a specific lipid called retinol. Okay, so we'll go over here. And at rest, your rhodopsin molecules and the retinol, or the composition of retinol chemicals in there is at this cis conformation. Okay, so it's shaped like this. But these are so sensitive that one photon can actually change this bond here from this kind of crooked to flipping it upside down so it is then straightens out that chain, essentially. So the difference between these two is only that one shift. That one bond is basically flipped from the cis to the trans. And if you don't know what that means, that's fine. You should learn it in chemistry. Okay, so once it's flipped into this trans configuration, it changes the shape, it interacts with the protein, that sends an action potential down the neuron, and then is collected in your optic nerve, and however many of those are going off is what forms an image in your brain. But in order to get it back to this, so you want to use it again, right? You don't want to just use it once and then be done. In order to get it back into that situation, into the cisretinal position, it requires an enzyme, and then it's flipped back, that bond is flipped back into the cis position to be used again. Okay? And this is in the rod, and rhodopsin is the pigment then, the photoreceptor, in the photoreceptor, which changes shape. The red, green, and blue one have slightly different pigments. They're all opsins, and those are activated at different wavelengths in the visual spectrum. Okay, so that's how you see it. That's how photoreceptors work.
So is rhodopsin just activated at any light spectrum?
Right. Yeah. Just needs a photon of light, and it'll flip. Yeah.
If it takes ATP to regenerate the cis configuration, could you get too tired to do it? Could you run out of ATP to switch it back?
I mean, I don't know about too tired. But you'd have to be really, really depleted for it to stop working. But yeah.
As a do it have
As in dead. Yeah.
yeah.
Or I guess blind, right? If it's no longer being used.
Yeah, but would you, because that's like photo bleaching where your eye's stuck in what you see, so it wouldn't be like you can't see anything, it's like an image is stuck in your eye.
Yeah, so photobleaching, yeah, that occurs, right, when you get too much light all at once, and then your brain picks up the opposite, so those ones that are bleached for now, yeah. But yeah, I don't think, there's always got to be ATP available for it to work, and so your brain is constantly supplying that. But, and you have a blood supply going to your eyes, constantly feeding that. One of the ways, so I don't know, do you guys know who Jack Kevorkian is? Dr. Death, not the Dr. Death and nothing. Yeah. So this is a guy who helped people commit suicide, assisted suicide in the 90s. He also developed a way to tell if someone was dead by looking at their eyes. And the way they did was he just looked at the blood vessels, and once the blood vessels stopped, then they were pronounced dead. So once they stopped pumping blood, essentially. So I think it's a conserved system where if you have energy available, it's constantly going to be going to your eyes.
It's one of those necessary functions that your body will give
Right.
ATP to always.
And once it stops, you're dead. Good question. All right, so yeah.
So I just have a question about.
All right. Again, you guys are familiar with a lot of these structures. We're going to add to what you know, just like we did with the photoreceptors. The large external part of your ear, which you learn as a kid, right? Called the pinna or the auricle. All right, and it funnels sound into your ear canal. Okay, ours don't work incredibly well, but other animals have much bigger ones, which can funnel a lot more sound into your canal there. And then where that sound is going is to your tympanic membrane or your eardrum. And that starts to vibrate with those sound waves. All right, and then I'm not very good at drawing these, but you have three bones here. Right, our ear ossicles, the incus, malleus, and stapes. And the stapes is the last one. And it sits against the, so around it, we're going to draw the oval window. And the oval window sits right up next against the beginning of our cochlea. All right. And then you have a smaller round window below it, which also, I didn't draw it correctly, but it lines up with your cochlea. All right, then you have this tube which actually goes then into the back of your throat or nasal cavity. The eustachian tube. Also known as the pharyngotympanic tube. All right, and this is helps you to adjust the pressure in your middle ear. All right, so this is our external ear. From the tympanic membrane to the windows, you have our middle ear. And then all of this is our inner ear.
So where's the line between middle and inner ear?
So the oval round window. Okay, so the cochlea is for the transmission of the sound. We have these semicircular canals. Which are for balance, help you know where you are in space. And we're not going to go through that mechanism, but that's also detailed in your book if you want to go through that, another sense. And then we have our vestibulocochlear nerve. And the vestibulocochlear nerve, half of it is the vestibular nerve, which pulls information from the semicircular canals, and the cochlear nerve, which pulls information from the cochlea, and then you go into your brain. Okay, so if you were to do a cross-section of the cochlea, okay, it would have this kind of three-compartment section. And the top part is called the scala vestibuli. And the bottom part is called the scala tympani. And the middle part is called the cochlear duct. And so what happens is after this sound is, enters, is funneled into your ear canal, transmitted or starts the vibration of the tympanic membrane, is amplified through those middle ear ossicles, and then causes fluid within the cochlea to be displaced as this wave sound, this pressure wave starts to go to different parts within the cochlea. So as it does that, it'll bulge at certain areas. So it'll cause like a bulge here. Kind of want it rounded. Okay, and then underneath this little funny thing here, I'll zoom in. This is called the organ of Corti. And what it has are these little cilia, okay, these little hairs, and they're suspended in fluid. Okay, and so when the pressure waves causes this bulge, it'll bulge there by the scala vestibuli, and it'll bulge on the bottom part into the scala tympani, and then those cilia will move. Okay, and as they move, that causes then an action potential to pass from there into the cochlear nerve. And where these along the cochlea are displaced, and thus where these action potentials are transmitted, is how you interpret those sound waves into, or those pressure waves into sound. Okay? Those cilia can be damaged. So if you are listening to really loud music, their ability to move can be dampened and or totally paralyzed. And that's how you lose hearing. Or one of the ways you can. All right, so this will make more sense as we talk about what is sound. All right, so sound is pressure waves. You take a fluid that's at rest and then you compress a bunch of that all at once, it makes a wave, right? All the stuff behind it then starts to move. Okay? Waves have these properties. So how far the wave goes up and down is called the amplitude. And that determines the loudness. And we measure that in decibels. All right. How frequently a wave passes, through from one trough to another, or from one peak to another, is called the wavelength. And that determines the pitch, okay, or the note, how high or low it is. And we measure that in hertz. Okay, H-E-R-T-Z, not H-I-R-T, yes. Okay.
It's not wavelengths, or actual centimeters and meters, and hertz is the frequency.
So hertz is the frequency. It's the number of times it passes a certain area per second.
Isn't a wavelength just the distance between peak and peak?
Yes. So if you have a decrease in wavelength, you have an increase in frequency.
Right, because sound travels the same speed.
Yeah. So hertz is the number of times it passes over per second. All right, so I'm going to... So if you have, I unraveled this cochlea. We have our three chambers there. And you have the organ in the middle here with the little cilia. Right? And what happens is at this point right here and at this point here, you're going to cause the cilia to move. And then your brain's going to interpret that as a low sound. Okay? And then how many of the cilia are moved is going to determine how loud it is. Okay? So if you have an increase in frequency, Okay, so I have another sound here. Let's do a different color. It's going to go here, 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 and here, and that's going to be interpreted as a different sound in pitch. And then how many of those cilia is going to determine how loud it is. So we can hear from under 1 hertz to up to 20,000 hertz. Okay, 20 kilohertz. Generally, as you get older, you lose the higher frequencies first. So just through usage and old age, you start to lose those. And so one of the things they do with a hearing test is they play different frequencies, and then you determine, if you have hearing loss or how bad your hearing loss is. Okay, so we're going to take one of these right now.